Good morning, everybody. My name is Lou Brown. I'm with Spectradyne, and thank you for joining our webinar this morning. Before we begin, just a couple things. If you do have a question during the presentation, please raise your hand in the Zoom meeting. You'll see on the participants panel, you'll see a thing that says raise hand, and just click on that. And then when we get to the end of the presentation, we'll start taking some questions. That's about all we have from here. So I show a little bit past 10. So I think we'll just get going here. Again, my name is Lou Brown. And at this point, I would like to turn it over to our presenter, Franklin Monson.
Okay, thanks, Lou. Yeah, we'll get started. I wanted to thank all of you for joining. I know you're probably quite busy, so we really appreciate you taking the time to view our webinar. As Lou said, my name is Franklin Monson. I've been with Spectradyne since its founding. I'll be telling you about our technology. We titled these slides "What's Really in Your Sample" because I'm not only going to hopefully convince you of our unique and important capabilities, but also shed some light on maybe some misleading information that we feel is out there about nanoparticle distribution. So let's get started. I will give you an overview of the technology and then some measurement examples on protein aggregation and extracellular vesicles. I want to motivate the discussion by actually taking a step back to larger particles. If you could imagine you have a precious formulation of diamonds, I think we'd all like to have a mixture of diamonds in liquid. That'd be great. You'd probably want to characterize that mixture very accurately. And so how would we want to do that? Probably we'd say, well, I want to measure each of the diamonds one by one, and I want to measure them directly. I want to get a really good measure of the value of this formulation. Now, we might not be dealing with diamonds when we talk about biotherapeutics or other precious mixtures, but those can be quite valuable too. So why don't we do the same thing with submicron distributions? It's a very simple question with a simple answer. The technology just has not existed to do the same kind of measurement with your precious nanoparticles. Spectradyne has solved this problem by implementing a resistive pulse sensing that's capable down to 50 nanometers. Now, many of you probably have heard of resistive pulse sensing as the Coulter principle. It's basically the same thing. Coulter counters have been around for many decades. Beckman Coulter obviously sells them, and they are very good for measuring larger particles like cells. The problem is when you try to miniaturize that technology to nanoparticles, which are obviously much smaller and usually much higher concentration, you can have various problems. We've overcome those challenges. We measure down to 50 nanometers. We can measure up to 10 microns. Actually, we recently raised that limit from 2 microns to 10 microns. It's a fast and easy to use technology with no real adjustable parameters. You can see an image of the instrument here. It's about 1 foot on the side. Sorry to our European joiners for the foot reference, but anyway, it's not very large and fits on a benchtop easily. We implement this technology through the instrument and with what we call our microfluidic disposable cartridges. So the technology is microfluidic RPS or MRPS, and you can see an example of our cartridges here in the lower left image. The cartridges are comprised of a glass substrate with a plastic block on them. This microfluidic implementation allows us to measure samples with a very small amount of analyte, only 3 microliters. So that's been proven to be an important benefit to some of our biotherapeutic customers. And I'll describe what goes on in here, but basically there's a nanoconstriction that does the analysis, but it's not only that. So those of you who are familiar with the Beckman Coulter counter or other implementations of RPS that are out there know that one of the problems is that you can get clogging or blockages as you're sending particles through the nanoconstriction that does the analysis. Well, we have engineered these cartridges to overcome some of those challenges. In the center middle here, you see a nice, pretty image of fluorescent particles flowing through the constriction, and I'll describe a little bit more what's happening there on the next slide. It's not just a constriction, like I said. To overcome some of the blocking issues, we We implement onboard filters in the cartridge, so there's a network of channels upstream of the constriction that greatly reduce the chance of any clog. And we also have a provision through our microfluidic architecture to robustly clear blockages when they occur, which is, rare, but it does happen sometimes. So let me go on to a description of the technology. It's basically a nanoconstriction through which we're flowing a fluid, and each particle that goes through the nanoconstriction gets a measurement. So we're measuring one by one, direct size and concentration. As the particle, so actually, I forgot to mention, we're measuring the electrical resistance across the constriction. All right, so you have to have a conducting fluid. That's one of the requirements of the technology. As a particle approaches the constriction, it starts to block the flow of ions locally going through that constriction, and you get an increase in the resistance that you're measuring. That resistance deflection is maximal when the particle is in the center of the constriction, and then as the particle leaves the constriction, the resistance returns to its baseline level. Now, let me spend a little bit of time on this because it's important to understand the information we're getting from this transit. We call this a transit. First, the size of this peak is proportional to the volume of the particle, or more specifically, the volume of occlusion of ions in that channel. Now, it may be counterintuitive, but this works whether or not the particle is conducting or dielectric. So you can have gold nanoparticles. We've measured gold nanoparticles many times. You can have silica, polystyrene, proteins, vesicles. So long as there's a blockage of those ions, you get a measurement. Implied in that is that we're insensitive to transparency issues. So you can have a biological particle that is basically, not detectable with light scattering in water, and we will have no trouble measuring it. So size of the spike is the volume or the size of the particle. The width of this transit or the transit time gives us information on the flow rate, and that can be used to extract a concentration. So each transit is giving us size and concentration information. When we start producing a family of cartridges, we call that a lot. Those cartridges are pre-calibrated at Spectradyne's facilities with 2 parameters that convert its measurements to size and concentration. So that's a cartoon. Let's talk about some actual data. So this is a mixture of polystyrene beads. Each peak has been highlighted with a red dot, and it's easy to see that there's basically 4 components mixed into the sample. We can plot that into a histogram format, and I do that kind of more conventionally here on the next slide. So those 4 components show up very clearly here. I want to point out some important information about this chart. So first, the x-axis is a linear scale. Those of you who are familiar with light scattering-based approaches often see a log scale on the x-axis, so it's really important to note that difference. We need to plot linear because we have such high resolution. So here you're seeing well-separated peaks, 4 of them, in only a 100 nanometer span, from 50 nanometers to 150 nanometers. Each peak has a mean size with it and a concentration number. So this is a concentration measured across that peak. There's a Gaussian fit here, which is nice to do. It's unnecessary, but we like to do it. And each component was prepared to be about 10 to the 9 particles per milliliter, and in fact, that's what we measure. So, I don't think this kind of information can be attained by any other method. Let me show you some more examples to drive that home a little bit more. So we're going to give you a few examples of our NCS1 measurement compared to DLS and NTA, which I think are 2 of the more popular measurement technologies out there. The information I show is also available on our website at the library link. So I'm going to show you 3 samples. The first is very similar to what we just discussed, polystyrene beads, and then we'll move to a more real-world example with a bacteriophage with and without polystyrene beads added. So first, let's build up this example. If you pretend you get a DLS measurement on some kind of formulation that looks like this in green, you would say, well, I got a peak in my distribution around 170 nanometers. in this view, it looks pretty broad. Actually, if you plot this in log scale, it looks pretty narrow. So great. We have a nice particle size peak. Maybe we want to get a higher resolution view, so we move to NTA. And maybe not surprisingly, the peak is shifted to the left. I think those of you who use DLS know that it often over-reports particle sizes just because it's so sensitive to the size of the particle and tends to get overweighted by particles in the tail to larger sizes. The NTA plot's a bit narrower, a bit shifted left, but it doesn't really show evidence of what's really going on in this formulation, and that's shown in blue here. And let me just say, we made the NCS1 measurement, but we sent this formulation out to an independent testing lab called Particle Technology Labs in Chicago. So they're a nice, capable testing house. They have NTA, DLS, and other technologies. So this is an independent comparison, and I think, again, illustrates the power of the technology in showing not only detail on the peaks, but what's going on in between the peaks. And of course, we can also get concentration information in each of these ranges. So that's a very artificial example. We use that just to kind of show some of the resolution capabilities of the technology. But let's go to a more real world sample that might be more interesting to some of you. Here we're measuring a bacteriophage centered around 62 nanometers in this blue region to the left. We also added some polystyrene beads at around 150 nanometers. So this gives us an in-measurement control. Just gives us confidence that everything's working correctly. The NTA and DLS plots, which are in red and green again, look like they are probably measuring the polystyrene bead, okay? But I can prove that to you by basically removing the beads. So that's shown here, all right? So we take the beads out of the sample. Our technology basically measures the same. So we have confidence that this is a good measurement, but we don't really need those beads. We measure the same kind of peak and the same concentration, 7 times 10 to the 9 particles per milliliter. In green, there is a DLS peak, but DLS will always try to give you a peak. In this case, the polydispersity index was quite high. I believe it was over 0.7. So a knowledgeable user of DLS would not trust that data. And then the NTA plot kind of collapses. These bacteriophage particles are just too transparent to be picked up at the small 60 nanometer size. One more thing I want to point out is, it's always nice to see this peak, gives us a good feeling that everything's working. However, to a researcher interested in this phage, they might actually have more information for what's going on off peak. So there's an asymmetry to this peak. There's quite a tail. Maybe the phage is dimerizing. Maybe there's remnants left over, or the purification protocol is not working very well. So I think sometimes the detail that one gains from I guess unexpected or unconventional views of the particles can be also informative. It's something to think about because this kind of information isn't really accessible through many other ways. So we basically are giving new information, and I think pretty powerful information. I'm going to move on now to some protein aggregation information. Again, this information is available on our website. Before I show you protein aggregation data, though, I want to point out a great paper that was recently published in a PharmRes journal. This is by Bristol Myers Squibb scientists. We had no role in collecting the data or in writing the article, but they did a comparison of our technology and conventional light scattering based approaches. And it's a good article for us. It shows some of the power of our technology. If you want an independent view of our technology, please go to this paper. I think you'll like it. So let's move on to the measurement. So what I'm going to show here is 5 stressed protein samples. These were measured on site at a biopharma company. It's a proprietary mixture, so we don't know exactly what did it, but we know it was in an acetate buffer with an ionic strength of about 20 millimolar. The stress time was varied across the 5 samples. We don't know exactly what the stress was. It's probably just temperature or heat. We measured on site, as I mentioned, without any additives. So we didn't run any We didn't add any liquid or conductivity or surfactant. We run neat. The conclusions we can draw from this measurement are a very clear quantifiable trend in aggregation as stress time increased. And we'll show you that earlier detection of the aggregation is really powerful and probably will be able to save people time and money. But here's the data. In blue is the control, and then we're varying stress 10, 20, 30, 60 minutes. You can see the 60-minute data shows quite a bit higher level of aggregation, so that's probably not surprising. So more stress, more aggregates. On the next slide, I want to show you a concentration trend in 3 different size ranges. So in blue, from 150 to 250 nanometers, we're going to call out the concentrations in these bands. And the point of doing this is to try to drive home that there's 2 benefits from being able to measure smaller particles. When you measure smaller particles, you effectively are measuring potential problems or stability earlier in the process, so you don't have to wait as long, right? Because the particles haven't needed to form as large an aggregate, okay? But, our technology not only can measure smaller particles, it can measure smaller shifts. So we get to look at shifts at a much higher resolution with a much higher resolution view, not just a higher sensitivity. So those are 2 separate things that are important. Let me just show you that, enough talk, right? So in blue is the 150 to 250 nanometer range. You can see that it's quite distinct from the green and red plots. So even at a 10-minute stress conditioning, we're measuring an important shift in the level of aggregate. All right, so this allows us to detect sooner, as I said, and to have more confidence. Now, we know that researchers won't be able to stop here. You have to do measurements in the subvisible range, so you're probably going to use HIAC or flow imaging or some other technology that measures supermicron particles. But you don't want to have surprises, right? As you go through your formulation process, you want to have confidence that stability is going to be good, and it's expensive to be surprised. So this gives the researcher an earlier indication of stability problems or the lack thereof, and that can save time and money. Let me give you one other protein aggregation example. This is one where we have a bit more of a comparison to NTA. Again, this is a stress protein formulation, in this case at 5 mg per mil. We ran the samples neat. What we show in this data are 2 cartridge ranges. So what used to be our largest cartridge going up to 2 microns, so 250 nanometers to 2 microns, and then a smaller cartridge that overlaps the larger one. It runs from 65 nanometers to 400 nanometers. We then compare this to NTA. We have to limit the size range because the NTA can't really measure above 500 nanometers. And as you see, its sensitivity will affect measurements below about 100 nanometers. But let me show you. So first is the data. Our large cartridge data is in red here. And then this orangish color is a small cartridge data. We have excellent continuity between the 2 cartridges. So our software will automatically stitch together data if 2 cartridge measurements are required. I want to point out that this is a huge concentration range, right? It's log scale on the y-axis, linear in the x. It's log scale on the y-axis because this is an 8 order of magnitude range, right? So I don't know if there's any other technology that can show such a large concentration range over this pretty broad size range too. I will point out that we do often see this shape from aggregation studies. So it is useful to have this concentration range and be able to measure over this span. Okay, now let's focus on the sub 500 nanometer range. So this is the expanded view. Again, our data is in the red and orange color. The NTA data is this yellowish trace. So first, you have to be careful. The NTA shows a loss of sensitivity below 100, 120 nanometers. If you didn't have our comparison data, then you may conclude that the data is good down to 60 nanometers and you actually have a peak. Maybe you think, oh, look, because remember, on a linear scale, this would be a very pronounced peak at 120 nanometers. But that would be an erroneous conclusion. In fact, NTA is underreporting the particles by about 10 to the 4 at 60 nanometers. In addition, there's some artifacts going on higher in the sizing range. So It looks like NTA is underestimating concentration in the smallest range and overestimating in the larger range. We see these oscillations commonly when we do measurement comparisons. I'm not an expert in the technology, so I think it probably has to do with the way the optics are arranged or maybe reflections in the flow cell. I don't know, but we do sometimes see this structure. Okay, so that's, I think, it for protein aggregation. For now, let me move on to another biological example of biological particle distribution. So this is extracellular vesicles, pretty hot area these days. Again, the information is posted on our website. Before I get into some of the detailed measurements, I want to just give a view of repeatability because, you know, this is something, it's probably the first question we get asked by ED researchers because they know they have broad particle distribution and they want to make sure that we can do basic measurements. In this case, we're measuring vesicles in a cell culture media from an in vitro fertilization protocol. And we measured 3 times on the same sample in 3 different cartridges, okay? And there's no extra scaling going on here. These are 3 measurements using 3 different cartridges, and you can see excellent repeatability from 2 microns down, in this case, to about 250 nanometers. Let me move on now to some more interesting information. This is a view of reference urinary vesicles, so measurements of pooled urine from 5 donors. The data I will be showing was actually presented at the annual meeting of ICEV, which is the International Society for Extracellular Vesicles. That was a meeting in last year, May, in Toronto. Our CEO, Jean-Luc Freiken, presented this data. We're going to show information from the NCS1, from NTA, and from TEM. So first, TEM, which is used as a reference. I think everyone knows TEM is a very powerful technology. It's difficult to use, it's slow, expensive, but if you're willing to take the time to do a careful TEM measurement, it's hard to beat it. In this case, the TEM measurement shows a clear power law distribution of these vesicles going down to about 50 nanometers, okay? So we're using that as the reference. Now we compare it to an NTA. This NTA plot is a bit more complex than the other ones I showed. In this case, 2 different gain settings are used to extend the range of that technology. In blue is 1 gain setting going up to 700 nanometers or so, and then at the higher gain setting, there's an attempt at least to extend the technology to a little bit below 100 nanometers. However, similar to what I showed with the protein aggregation example, the NTA tends to underreport the population of particles in the lowest size range. There are some oscillations or false peaks as you go to higher size range, and basically a poor fit to the TEM distribution. So with the aggregation, I kind of use the NCS1 as a reference and compared NTA to that. But you know, we're a new technology, so this is interesting because I think we're comparing to a more robust proven technology, but drawing kind of the same conclusion. All right. Now, if we look at the NCS1 data and compare it to this distribution derived from TEM, we see excellent agreement basically between the 2 distributions. Our measurement is, again, 2 cartridges, so a larger cartridge in blue, a smaller in black. In this case, we spiked in our control beads, again, to just have a nice in-measurement control. I don't want to dwell on that too much. However, I do want to point out that this is a pretty powerful thing to be able to do. Because we're measuring particles independently, we can add, say, 500 nanometer particles and not affect at all what we measure at 100 or 150 nanometers. Whereas with light scattering, you might get some correlation effects and corrupt your signal. This can be important because maybe you're looking at a population of EVs in a purification protocol. You're also, though, concerned about whether there's contamination or remnants at higher or larger particle sizes. You'll get independent assessments of those 2 things with our technology. Anyway, so you see the peak from the beads, but we get very good agreement lower down. Again, basically down to 50 nanometers. So accurate quantification of these reference EVs. Now, I've shown you a few times, and I've dwelled on it a little bit, this issue of spurious peaks with light scattering-based approaches. Now, so that's empirical, that's great. You can also model this theoretically, and we did a pretty simple example of this. It's posted in TechSpeak on our website. We'd be happy to talk about it with you more. But basically what we did is we mocked up a distribution shown here in blue of kind of a mock nanoparticle formulation. And then we simulated what NTA might measure based on the fact that the light scattering intensity varies as the 6th power of the particle size, shown in this formula here. So it's a relatively simple convolution to do. And if you do that, you see that this blue distribution gets transformed into this orangish one here. So you got to be careful. If you just blindly measure NTA and assume that the data is good to 0 nanometers or 20 nanometers, you will maybe be led to conclude that there's a peak around 50 or 70 nanometers. That would be an erroneous conclusion. We see this all the time from EV researchers and to some extent with aggregation researchers. So we'd love to prove this to you in more detail, but we do think that there's quite a bit of misleading conclusions out there from not understanding this phenomenon completely. Let me leave you with one more example. This is really more of an instrumentation study, but I think it's pretty nice data. So I'm going to show you measurements of biocomposite nanoparticles. So these are actual kind of drug delivery vehicles used by a nanomedicine company. So I've shown you aggregation, protein aggregation measurements, some EV measurements. Now we're going for some synthetic nanoparticles from a nanomedicine customer. This dilution series is from 10 to the 10 to 10 to the 11 particles per milliliter, so a higher concentration range than the others. The nanomedicine folks like to live at a higher particle density or particle population concentration. We did still add the 150 nanometer controls, and you'll see why. It shows a pretty powerful result. Measurements across 5 cartridges. So here's the data. The biocomposite nanoparticles are here around 60 nanometers. You can see a clear trend in concentration increase. At the same time, we spike in the 150 nanometer beads at the same concentration each time. So you're really getting 2 results from this set of measurements. One, the repeatability of the technology, which looks good, and the concentration linearity. How linear is it? Well, if we plot the measured concentration versus the expectation, we get a very nice line here. So I think this is a great example of what we can do with our technology. Okay, so that's pretty much it for the measurement examples. Just to summarize, the NCS1 measures particles independently, one by one. We can get size and concentration directly on all material types. It's fast and easy to use, no adjustable parameters, quite a small sample volume. We'd love to talk about your application with you. You can get more information on our website. We can do sample measurements if you're willing to send something out. We also often do demos on site. So that's it. I guess, Lou, we can open up for questions.
Yeah, great. Thanks very much, Franklin. And as I said at the beginning, if you have a question, just click on the raise hand button there and I'll unmute you so you can ask the question. I guess that's it then.
Okay, well, thank you for your time.
Yeah, thank you all for joining us today, and look forward to hearing from you and hopefully measuring some samples and talking with you further in the future. Thanks very much.